Recombinant DNA technology is the joining together of DNA molecules from two different sources. The recombined DNA molecule is inserted into a host organism to produce new genetic products in large quantities. Methods used in generating recombinant DNA are central to many contemporary areas of modern biology and medicine. Today, we will cover two topics. First, we will look at DNA cloning. Cloning is making an exact copy of any substance. DNA cloning or gene cloning is a way to isolate a specific DNA fragment and produce it in a pure form in large quantities. Here is a eukaryotic chromosome. These are eukaryotic genes. DNA cloning links eukaryotic genes to small bacteria or phage DNAs and inserting these recombinant molecules into bacterial hosts. DNA cloning requires 5 components. First, it requires the gene in the form of a DNA fragment. It can be extracted from biopsy or from a blood sample. Second, restriction endonucleases. They are molecular scissors. These are restriction endonucleases. Third, DNA ligase. Fourth, a carrier molecule or vector. Fifth, a living copying machine, a host organism, usually a strain of bacterium. Restriction endonucleases have several features. They cleave DNA in a sequence-specific manner. They are evolved as a defense mechanism against infection by foreign DNA. At the same time, host DNA is protected by methylation. Different restriction enzymes are found in different organisms. Here, we will use EcoR1 as an example to illustrate 3 other characteristics. First, each restriction endonuclease has a unique name. Here we use EcoR1 as an example. The first capital letter represents genus. The next 2 lowercase letters represent species. R represents strain. 1 represents the order of identification in the bacterium. Second, many restriction endonucleases recognize palindromic sites. For example, here, the top strand reads GAA-TTC. The bottom strand from 5' to 3' also reads GAA-TTC. EcoR1 cuts these sites at the staggered position, leaving overhangs. They are called sticky ends. Third, EcoR1 will not cut at these sites if adenine is methylated. By doing so, it protects its own DNA from being degraded. In bacteria, cytosine can be methylated at C5 or N4 positions. Adenine can be methylated at N6 position. Foreign DNAs which are not methylated are degraded by sequence-specific restriction enzyme. Bacterial DNA is protected by methylation. The methylation of native DNA acts as a primitive immune system. Here are specific sequences recognized by different restriction endonucleases. For example, EcoR1 only cuts GAA-TTC between G and A. KPN1 cuts GGT-ACC between 2 Cs. A 6-base pair cutter, such as EcoR1, will generate DNA fragments averaging 4,000 base pair in length from a genomic DNA. The frequency of cuts decreases in the genome when the recognition sequence is longer. Some restriction enzymes, such as TAC1 and EcoR1, form staggered cuts that produce sticky or cohesive ends. For example, TAC1 cuts between T and C. The resulting DNA fragments have single-stranded sequences that are complementary to each other. Other restriction enzymes, such as HEY3, produce fragments that have blunt ends. They are double-stranded. They do not form hydrogen bonds with each other. DNA ligase joins sticky ends. Ligase from bacteriophage T4 can covalently join the blunt-ended fragments, such as fragments generated by HEY3. As an example, here is a 6 kb DNA fragment with 2 EcoR1 sites. Restriction of this DNA by EcoR1 yields 3 fragments. The smallest fragment is from 5' end to the first EcoR1 site, which is 0.7 kb. The second fragment is from the second EcoR1 site to the 3' end, which is 2 kb. The third fragment in between 2 EcoR1 sites is 3.3 kb. On a DNA gel, 3 bands appear from the DNA cut by EcoR1. Using the same method, you can find out bands from DNA cut by other enzymes. Vectors function as DNA carriers to allow replication of recombinant DNAs. Typical experiment uses one vector plus a piece of foreign DNA. Foreign DNA doesn't have replication origin. It depends on the vector for its replication. There are 2 major classes of vectors. One is plasmid, and the other one is phages. Here are various vectors and size of the inserts they carry. Plasmid is the smallest vector. It carries up to 15 kb of inserts. Bacteriophage lambda carries up to 25 kb. Bacteria artificial chromosome carries up to 300 kb of inserts. Yeast artificial chromosome carries up to 2000 kb of inserts. Except yeast artificial chromosome, all other vectors can be propagated in E. coli. Here, we will use Plasmid PBR322 as an example to look at the structure of a plasmid. This vector is derived from a naturally occurring plasmid. It has 2 antibiotic-resistant genes, ampicillin-resistant and tetracycline-resistant genes. It has unique restriction enzyme cleavage sites. It has an origin of DNA replication. Here are steps of DNA cloning. The first step is generation of recombinant DNA. This is done in the test tube. Gene X is cut with PST1. The result is a DNA fragment with 2 sticky ends. Plasmid vector PBR322 is also cut with the same enzyme, which yields a linear DNA with 2 sticky ends. The vector and the gene X fragment are mixed with T4 DNA ligase. Ligation yields recombinant plasmid that contains gene X. The next step is transformation. The second step is transformation. The recombinant plasmids are mixed with competent bacteria that are capable of taking in plasmids. The third step is growing the bacteria on the solid medium, which contains antibiotics. Bacteria containing plasmids with antibiotic-resistant genes will grow on the medium. The others die. Therefore, only transformed bacteria survive. There are three ways to transfer gene to bacteria. They are conjugation, transduction, and transformation. As we know, transformation is transfer of a foreign DNA directly to bacteria. Conjugation is transfer of DNA from bacteria to bacteria. Transduction is transfer of DNA from bacteriophage to bacteria. This can be summarized in a mnemonic. CTT represent conjugation, transduction, and transformation. A genomic library contains clones of all genes from a genome. Genomic DNA is cut with a restriction enzyme, for example, SAL3A. At the same time, bacteriophage is cut with BAMH1. Ligation using T4 DNA ligase leads to insertion of each fragment into the phage. Recombinant phages are assembled using an in vitro phage assembly system. Then use these phages to infect bacteria. Each bacteria cell contains only one vector. Once a library is established, it can be used to search for any gene of interest. How do we identify the genes of interest? The answer lies in probe and hybridization. A probe is a short piece of single-strand DNA labeled with a radioisotope, such as P32, or with a non-radioactive molecule, such as biotin. The sequence of a probe is complementary to a sequence in the DNA of interest, called target DNA. A clone in a library that contains the target DNA is identified through screening. Screening is carried out using hybridization. This is the example of hybridization. Here, the probe is a single-strand DNA. It adheres to the target DNA and forms a hybridized double-strand DNA. To facilitate annealing between the probe and the target DNA, DNA samples are denatured by heating to 95 degrees. After radiolabeled probe is added to the sample, temperature is dropped to 65 degrees Celsius. This will allow the annealing of the probe to the target DNA. Radiolabeled probe hybridized with target DNA can be detected using an X-ray film. To identify a clone from a genomic library, we place a disk of absorbent paper or membrane on top of a solid medium on which bacterial colonies grow. Peel paper from the petri dish. Now the paper contains bacterial colonies. Lyse bacteria, denature DNA, and perform DNA hybridization using a radiolabeled probe. At the end, expose the paper to an X-ray film. Position with strong radioactivity corresponds to positive clones. cDNA is the abbreviation for complementary DNA. It refers to an mRNA transcript sequence expressed as DNA bases rather than RNA bases. cDNA is derived from mRNA. As a result, it contains only exons, but no introns. A cDNA library is a set of clones representing as many as possible of the mRNAs in a given cell type at a given time. To construct a cDNA library, we first isolate and collect mRNA, followed by synthesis of cDNA using reverse transcriptase. Double-stranded DNA is then inserted into plasmid, transform bacteria using recombinant plasmid, grow the bacteria, and finally verify the sequence by DNA sequencing. Among these steps, the most critical step is synthesis of cDNA. Reverse transcriptase cannot initiate DNA synthesis without a primer. Typically, oligo-DT is used as a primer that complements poly-A tail at the 3' end of an eukaryotic mRNA. After the first DNA strand is synthesized, RNA is degraded using RNase H. Then complementary DNA strand is synthesized using DNA polymerase. Leftover RNA fragments are used as primers. DNA cloning has many applications. It has been used to produce recombinant therapeutic proteins. It also been used for genetic diagnosis, for mapping and identifying disease-causing genes, and for gene therapy. It has been used to generate transgenic and knockout animals. For example, several recombinant therapeutic proteins have been produced using E. coli as hosts. These include insulin, growth hormone, and epidermal growth factor. This is insulin. Here is the genomic DNA around beta-globin gene. In a normal allele, there are 3 MST2 sites. As a result, after restriction by MST2, we see a 1.15 kb and 0.2 kb bands. But in the sickle cell allele, second MST2 site is destroyed. As a result, we only see one band, which is 1.35 kb band. In this family, both parents are carrier. We see both 1.15 and 1.35 kb bands. In the third child, we only see 1.15 kb band, indicating this child is homozygous for the normal allele. DNA from the fetus shows 2 bands. This result indicates the fetus is heterozygous for the sickle cell allele and is a carrier. Gene cloning has been used for gene therapy. For example, severe combined immunodeficiency disease, or SCID, is caused by deficiency either adenosine deaminase or a gene coding for interleukin receptor subunit. For example, this patient has SCID caused by adenosine deaminase deficiency. To treat this patient, lymphocytes are collected from the blood and the normal genes for adenosine deaminase is transferred to the lymphocytes using virus-mediated gene transfer. Lymphocytes are then returned to the patient, and the patient regains immune function. This type of gene therapy is called gene replacement therapy. Finally, gene cloning has been used to generate transgenic animals. Transgenic animals can be produced by injecting a cloned gene into the fertilized egg. If the gene becomes integrated into a chromosome, it will be present in the germline of the resulting animal and can be passed along from generation to generation. Now, let's move to part 2. We will look at tools in genetic analysis. The first tool is Southern blot. Southern blot is used to identify specific DNA fragments. More specifically, it is used to detect copy number of gene. It is also used to detect gross alterations in the gene. It allows for visualization of specific DNA fragment among thousands of molecules. It can also be used to detect more than one fragment of DNA. Procedures for Southern blot can be better illustrated in the next slide. DNA samples are digested with a restriction enzyme. Digested DNA samples are separated on an agarose gel. Separated samples are transferred to a filter by diffusion. The filter is treated with alkali to denature DNA. Resulting single-strand DNA binds to the filter. Add the labeled DNA probe to the filter. Hybridization begins. Rinse away unbound probe. Use an X-ray film to detect bands. Southern blot has been used in determination of restriction fragment length polymorphism, or RFLP. RFLP is a variation in the length of restriction fragments produced by a given restriction enzyme in the DNA sample. Alleles are distinguished by the size of bands obtained by Southern blot analysis. For example, person 1 has an allele with 100 ACT repeats. Person 2 has 400 ACT repeats. These two alleles can be distinguished by cutting their genomic DNA with EcoR1, followed by Southern blot analysis. Northern blot is a variation of the Southern blot. It is used to detect RNA instead of DNA. Here are bands detected from Northern blot, showing a particular RNA transcript is differentially expressed in different tissues. Western blot is used to detect proteins. Similar to Southern blot, protein samples are first separated by a gel and then transferred to a filter. Different from Southern blot, a polyacrylamide gel is used to separate proteins. Antibodies are used to detect specific bands. This is a result. Each of these bands represents a protein. Polymerase chain reaction, or PCR, is in vitro cloning. It is a means of selectively amplifying a particular segment of DNA. The most critical parameter for successful PCR is the design of primers. Here is the target DNA. After two strands are separated, the primer that primes DNA synthesis from left to right. It's called forward primer. The one that primes DNA synthesis from right to left is called reverse primer. Three key components for PCR reactions are template DNA, a pair of primers, and heat-stable DNA polymerase. Other reagents needed include deoxynucleotides, magnesium for DNA polymerase, and reaction buffer. Here are steps in PCR. First step is DNA melting. The reaction chamber is heated to 94 degrees for 30 seconds. This causes DNA melting. Two strands are separated. Second step is annealing. The reaction temperature is lower to 50 degrees Celsius for 30 seconds, allowing annealing of the primers to each of the single-strand DNA templates. Third step is extension. The reaction temperature is raised to 72 degrees Celsius, optimum activity temperature for the DNA polymerase. DNA polymerase synthesizes a new DNA strand by the end of the extension step. The number of DNA target sequences is doubled. The processes of melting, annealing, and extension constitute a single cycle. We are ready for the next cycle. Typically, a PCR reaction is set for 30 cycles. Here is one same reaction run with different number of cycles. More cycles used for reaction, more DNA will be generated. PCR plus electrophoresis on agarose gel can be used to distinguish two alleles with a difference in DNA length. PCR has a wide range of applications. Here is one example. PCR is used to detect short tandem repeats. A short tandem repeat is a region of DNA containing tandem copies of di, tri, or tetranucleotide repeat units. For example, GT represent dinucleotide repeats. ACG represent trinucleotide repeats. Short tandem repeats are also called microsatellites. Number of repeats vary greatly between individuals. Alleles are distinguished by the size of the bands obtained by PCR. There are 13 forensic short tandem repeat markers currently used by FBI. DNA sequencing is a gold standard to identify point mutation, small deletion, or insertion. Double-stranded DNA is used along with many copies of a primer that binds only to one strand, often the coding strand. A sample of DNA to be sequenced is put in each of the 4 reaction mixtures containing a DNA polymerase and all the necessary dNTPs required to synthesize new DNA. In each test tube, a different dideoxynucleotide triphosphate, ddNTP, for example, ddATP. It lacks both the 2' and 3' hydroxyl group. These ddNTPs can be inserted into a growing chain of DNA, but then any subsequent elongation is stopped. Pieces of newly synthesized DNA in each tube are separated by gel electrophoresis in 4 different lengths, as shown here. The sequence of the new strand can be read from bottom to the top. The sequence of the newly synthesized DNA strand in this example is CTT GGAAC TGTA, as shown here. Reverse transcription PCR, or RT-PCR, detects and can quantify a specific RNA rather than DNA in the sample. This test is useful in detecting RNA viruses such as HIV. A blood sample from HIV-infected individuals is obtained and is treated with reverse transcriptase to produce cDNA from any RNA in the sample. The cDNA is subsequently PCR amplified using primers specific for the end sequences of the HIV cDNA. The amplified product is quantitated and, with the use of standard curve, can be related to the original amount of HIV RNA present in that patient. This whole process is summarized in this chart. A DNA microarray, also known as DNA chip, is a collection of microscopic DNA spots attached to a solid surface on a slide, as shown here. Each spot contains picomoles of a specific DNA sequence, known as probes. Here are probes. Sample can be cDNA or antisense RNA. For a typical experiment with microarray, first step is to isolate 2 RNA samples from 2 different sources. Second step, RNA samples are used as templates to synthesize cDNA. Third, 2 cDNA samples are labeled with 2 tags in different colors, red versus green. Fourth, cDNA samples are hybridized with a microarray. Fifth, specific color from each spot is recorded using an imaging system. When the gene from sample A is more abundant than that from sample B, the spot will appear to be red. Vice versa, when the gene from sample B is more abundant, the spot will be green. When their expression levels are equal, the spot will be yellow. Therefore, color of each spot reflects ratio of RNA levels of that particular gene from these 2 samples.